Okay, so good morning, everybody, and thanks for being here. So as Kavita said, I will give some lectures here on the topic of fitness landscapes. So it seems to me a bit loud. Is it okay, or it's okay? So let me start by sort of giving you an outline. It turns out when I started seriously preparing these lectures, I realized that there might be a bit too much material, so I might have to cut things short a little bit at the end, but we'll see that as we go along. So the overall title is Fitness Landscapes in Theory and Application. And of course, the concept of fitness landscapes is rather old. So it was initiated in the form that we're using it today in the 1930s by Sewell Wright and others. I'll get to the history a little bit tomorrow. But this topic has received a lot of attention over the last 10 or 15 years, and there's a lot of new data. And along with that, also new theoretical concepts and mathematical concepts have been developed. And this is what I want to convey to you. So although I'm a theorist, as Kavita said, so I'm fond of doing mathematics and theory, I really also want to emphasize the applicability of these ideas. So the first lecture, which I will do today and probably part of tomorrow, is called Fitness and Epistasis. So these, of course, before talking about fitness landscapes, we have to explain what fitness is. Both of these are important, but also somewhat elusive concepts in population genetics. And so I think it makes some sense to talk about what we actually mean by them. And my point of view as a theoretical person is that things become clearer if you try to formulate them mathematically, right? So that's what I'll try to do. The second part, this will be mostly, so today I'll be working mostly at the blackboard. Tomorrow I'll be showing some slides, and there in particular I want to show you a lot of empirical examples of fitness landscapes. So this is the second lecture, Empirical Fitness Landscapes. And then the following two lectures will be addressing models. So given that we have data, the question is how can we describe them, how can we interpret them, and this is where mathematical models come in, and there are sort of two flavors of models that I want to discuss with you. The first ones are, the first type are probabilistic models, which means that you, as we'll see, fitness landscapes are sort of defined on a certain space, and so we give a kind of probability measure on that space, and generate realizations that have certain properties. The second flavor is probably a bit more biologically motivated, and this is a kind of fitness landscape where we actually try to introduce an explicit phenotypic level. So here we're going to think about what happens if we relate genotype to phenotype and then to fitness. So these are genotype, phenotype fitness maps. And the most famous example of such a map is called Fish's geometric model, which some of you might have heard about. And finally, the last lecture, but as I said, I may not actually get to that. I call it Navigating Fitness Landscapes. So here the topic will be, once I have a fitness landscape, what is the dynamics of a population evolving on that fitness landscape? And this, I think, might also tie up a bit with some of the things that Daniel will be doing. Okay, but I'm not under pressure to cover everything. I gave you in the online material there are a number of papers where you can read about these things if I don't get to explain them fully. So the most important thing here for me is that we're able to communicate. So as Kavita already said, do ask questions, and as Kavita has taught me, if you don't ask me questions, I will ask you questions. So you have to think about what is better, right? Okay, what you prefer. Okay, so any questions so far? Okay, so let me start with the first topic. So let's first think about fitness. So fitness, of course, is a very a central concept in evolutionary biology and population genetics. Now, why is fitness important? So from my point of view, the most important aspect of fitness that we're going to deal with here is that fitness determines how gene frequencies of populations change by selection, right? So this is sort of the reason why we're interested in this. So this is a kind of determinant of the dynamics. So fitness governs how gene frequencies change by selection, of course, together with other factors like genetic drift and so on. But this is really the most important part. Now, the question is, how do we describe this mathematically? There are sort of two levels on which I want to discuss it, and I'm sure that this is something that all of you already know, but I think it's important to be a bit explicit in order so that everybody's on the same page. So there's sort of a deterministic view of selection, where you would say, okay, let me write down some equation for the gene frequencies, right? So let's suppose we have some types. So think about genetic types or phenotypes. It doesn't really matter at this point. We have some types which I label. So I label them from 1 to k, and these occur in the population at frequencies xi. And they have some fitness values, and I want to see how these frequencies evolve in time. Okay, so the simplest description that you could write down would be a continuous time description, or what I want to emphasize here is that you can describe these things in continuous time or discrete time, and this will play a certain role a little bit later. So I want to make this distinction. So let's first look at the continuous time case. So then we would write down a differential equation for these frequencies. And this differential equation in the simplest case would look like this. So the gene frequency of type A would grow if the fitness is larger than the mean fitness of the population, and it would shrink if the fitness is smaller, right? So this would be the simplest kind of situation. So here the fitness values, and so this F bar is the mean fitness of the population. Right, so this is Fi xi. So if Fi is greater than F bar, these genes will increase in frequency. If it's smaller, they will decrease. And these Fs here are called, or I will call them here, Malthusian fitness values. Right, so this is a particular version of fitness that we will be using here. And you can think about, you can think about this, for example, for two types. So if you want to, get more familiar with this, you can think about this problem for two types and try to solve it. It turns out that these equations can actually be solved explicitly as long as these Fs here are just constants. You can solve them for any number of types. In this one of these lecture notes that I posted online, this is also posed as a problem. Okay, so that's sort of one version, but we can also do the whole thing in discrete time. So if we think about generations. Yep. Yeah, so I'm keeping this very general. Think about, for example, a certain gene that comes in different versions, different alleles, right? So, or think about people with red eyes or not red eyes, blue eyes. But at this point, I don't have any more structure than that. I know that there are different types of individuals in my population, and they differ in their fitness. This is all I really need to say. Okay, so this is. Later, we'll talk about genotypes and phenotypes, but at this point, this is just a way of, think about red and green individuals or whatever. Okay, so if we think about this in discrete time or in discrete generations, then we could write something like this. So the frequency in population t plus 1 is obtained from the frequency by selection again in population T by the fitness divided by the mean fitness multiplied by the present frequency. And again, this W bar then is the mean frequency, mean fitness in the population. And this Wi I call Wrightian fitness. So both of these, of course, are very commonly used in population genetics. And the reason why I'm sort of mentioning them here is that this will become play a certain role later if we define epistasis. Because these, mathematically speaking, these two equations have different properties of invariance. So here you can see that if you added some constant to the fitness of all the individuals, nothing would change, right? Because here we only have a difference between fitness values. And similarly here, if you multiply the fitness of all the individuals by some constant, again, nothing would change, right? So the point that I want to make here is that in continuous time, we have, okay, maybe I should write it on the next blackboard. I still have enough space. So, so the continuous time dynamics is invariant under additive changes of fitness. And sometimes we say that here fitness is measured on an additive scale. And the discrete time dynamics under multiplicative changes. And because there is a certain amount of confusion in the literature as to which scale is the right one. I don't think there is any scale that is the right one. You just have to know what you're doing. Yes, please. Excuse me? Yeah.
Between these two fitness measures, that Fi and Wi?
Well, in some sense, they describe the same thing. I mean, the way you could think about this discrete time dynamics mathematically is that you take your continuous time dynamics and you integrate it over one generation, right? So then you have something that is discrete in time. What I just want to emphasize here is that I mean, biologically, I guess what people typically would call this is a model with overlapping generations, which means that individuals sort of reproduce, at any time. So the change from one generation to the next can occur at any time, whereas here, in some sense, all the individuals reproduce at the same time. So this is often mentioned as a model for something like annual plants, which basically reproduce simultaneously. What I just want to emphasize here is that this has this, naturally has this additive structure, and this has this multiplicative structure.
Doubt I had, I mean, in FI, you can say that is the fitness of an individual, right? I mean, F1 is the fitness of first individual.
Oh, type one.
Yeah, one genotype,
an individual carrying that type, yeah.
So then what you will tell about W1? Is it
WI is also the fitness of individual I.
Of an individual.
Yeah, it's the same. I mean, these are just, I mean, yeah, so, and you can sort of see that you can go from one to the other essentially by taking the logarithm, right? So both of these are fitness values associated with the types. Excuse me? No, well, it's just, I mean, you know, so suppose I have a generation time delta t, right? And so if I integrate this equation over one generation, then I would just get a relation like Wi being e to the fi times delta t. Right? So this way they would be related. Yeah.
Mathematical part of this, but I'm still trying to figure out how this fits in the biological system when you talk about the invariant under additive changes of fitness, or like the discrete dynamics is invariant under multiplicative changes. How does it actually fit into the biological system? I mean, like for an example, if you can.
Well, yeah. So let me take an example. So how do how would you measure fitness, right? I mean, this is of course what it really comes down to. So nowadays, of course, when people do evolution experiments with bacteria, you can measure fitness in a competition experiment, right? So you take your two types, you compete them against each other, and you see how one of them grows relative to the other. And so in a competition experiment, so what you typically would do in a competition experiment is that you would do serial dilution, right? Serial dilution, and then you would, so this means that you sort of dilute your population once a day, and so that would be a natural setting where you could apply a kind of discrete time picture, right? So you just look at the frequency from one day to the next, and then you expect that if one type has higher fitness than the other, every day the frequency would be multiplied by some factor. Okay, but you can also do a competition experiment, of course, in a kind of what are they called? Chemostat, right? So you put your population, you put your bacteria into a system where there's some dilution flow, so that they are kept at essentially constant population size, and they just there's a continuous flow of nutrients and waste and so on. And then you would probably like to do this use a continuous time setting instead. Right. So this is maybe one way to think about that. And in both cases, you can sort of measure what the fitness is, right? Yes. Continuous time means this. So continuous time means that the frequencies. The frequencies change, yeah?
The example just now you gave, right now, for the continuous one.
Chemostat, yeah.
Yeah, chemostat, yeah.
Chemostat. so I'm not an experimental evolutionist, but as I understand it, you have a reactor where you have a flow of nutrients and you remove waste products at a constant rate in such a way, and you sort of, for example, there's this famous what is called the morbidostat, right, which is a system that was invented by someone in the US to study the evolution of antibiotic resistance. So say you measure the optical density of your solution, and you have a feedback mechanism, optical density constant, right? So as if the population density increases, then you increase the flow rate so that things get sort of flushed out more quickly. And that way you can maintain essentially a constant population size, and bacteria are sort of constantly dividing and being flushed out of the system, right? So that's sort of the rough idea. Okay. Okay, but I see I didn't really want to make such a big point of this, but maybe it's good to mention it at least at some point. But so the main thing that you should sort of keep in mind is that there are different scales on which you can measure fitness, and but I think these scales are not, there's sort of nothing intrinsic about them. It's largely determined by by your experimental setup or by the nature of the populations that you're studying. Okay, and so here, unless I state otherwise, I will always be thinking in terms of Malthusian fitness, which is additive. Okay, so here we will typically adopt the Malthusian picture. Okay. So this is sort of the deterministic view of what fitness does. There is also a kind of a different, but of course related context in which fitness plays a role, which is sort of stochastic or related to genetic drift. And this is when you ask, when you have a mutation appearing in a population and you ask for the probability that it fixes, right? So this is sort of the process of fixation. So the probability for a mutation to fix in a population depends, of course, also on its fitness, or more precisely, it depends on the fitness again relative to the resident population, which is what I will call here the selection coefficient. And so the selection, and because I'm sort of adopting here the additive scale, as I said, the selection coefficient is a difference between the mutant fitness and the wild type fitness. So wild type here simply means a population into which this mutant occurs, right? So this is sort of the original type, if you will. And so in the context of genetic drift, the way fitness manifests itself is that if you have a mutant with higher fitness, it will be more likely to fix. There's some formula for this fixation probability, but we actually don't really need that at the moment, so I will not write it down. We'll get back to that when we need it. I just wanted to mention that in a sort of probabilistic setting, of course, fitness has the role of sort of changing the chances for fixation events to happen. Okay. Okay, so that's sort of my working definition of fitness here is, you should always have in the back of your mind a kind of equation like this. And so these would then be the selection coefficients. So now we can call these selection coefficients. So this is now the selection coefficient of this particular type. And what we're going to talk about next, and this is sort of the topic of epistasis, is how the selection coefficients of multiple mutations are related to selection coefficients of individual mutations. Okay. Yeah. Microphone.
I was just wondering, like, so we usually consider drift as a very stochastic process without any directionality. So how will account a mutation, like, in a stochastic process, how the selection coefficient is valid there? I'm just curious.
Okay, so since you're asking, I think I should write down the formula after all. So what I mean here, I guess, the meaning of genetic drift is maybe also a little bit vague. I guess you're right, if you just have genetic drift, this is a stochastic process. What I'm talking about here, if you will, is the interplay of genetic drift and selection. And the way this sort of manifests itself mathematically is, for example, through this famous Kimura formula for the okay, but let me just let me just write it down. So the Kimura formula for fixation, so for the fixation probability. So this is the probability of fixation for a mutant that is initially present at some frequency x, right? For a mutant initially present at frequency x. I like to call this pi. I don't know what you will call it, maybe not pi. But the point is that this is a function of the selection coefficient of the mutant, its initial frequency, and the population size, right? So this is this famous formula. And so okay, so this looks now a little bit complicated, but what you can see is that the probability will be larger if x is larger, but will also be larger if s is larger. So having a larger selection coefficient will enhance the probability of fixation. If s is negative, then this will be very small, so deleterious mutations don't fix. And of course, the case of pure genetic drift that you refer to here is taking the limit s to 0, which you can do in this formula, and then you see that the the fixation probability is simply equal to the frequency, right? But this is probably what you would refer to as genetic drift, but this formula sort of expresses how selection interacts with genetic drift. Okay? Yeah.
Diploidy? The 2 in the formula?
No, the 2 is the 2 comes from the diffusion approximation. I mean, so the 2 is from the Wright-Fisher model, I should say. I mean, this factor changes. This factor here depends a little bit on what precise model you use. But the diploidy would give you another 2. So if you have diploids, then you should replace the n by 2n. Excuse me? Yeah, I mean, this is sort of a mathematical statement within a certain model, and this model has a fixed population size, so it is a population size. I mean, I should also say there is no, there is no mating. These are, everything I'm going to say or everything I've said so far is thinking about asexual populations, so there isn't really any effective population size in that sense. Well, you can use it for the effective population, but as a mathematical statement, it's about a model with a fixed population size. I mean, people use it within effect, but Maybe Daniel will devote a lecture to the effective population size, I don't know. Right. Okay. Very good. Any more questions? Okay, so let me get to the.
I have a question. So in your continuous time,
there you are. Yeah.
There is the assumption that your average frequency of all the phenotypes in your population is constant with time. So Fi minus F bar. So F
it's
bar.
not constant. No. This of course also changes. I mean, what happens under this dynamics is that the average fitness changes, and so therefore more and more of the types will no longer grow. And in the end, the type of largest fitness will dominate under this dynamics. So in that sense, maybe this SI is not a very good, is actually, yeah, so forget about this. This is not really what I mean by I mean, I typically mean selection coefficient with respect to a fixed background.
Right.
Okay, thank you. All right. So. Okay, so now let's think about epistasis. So this term historically was first introduced by Bateson in 1908. And so where does this word come from? So it's made out of Greek parts, and what it actually stands for is, if you translate it literally, standing upon. So one thing is sort of puts itself on top of another and blocks it. And so what the original definition of Bateson was of epistasis was that the effect of a certain mutation is blocked by the presence of another one, right? So you have a mutation that makes your rabbit black, but a second mutation keeps it white as it was initially, right? So this is sort of the classical meaning. Blocking or masking of the phenotypic effect of one mutation by another. Now, in sort of modern use, or at least the way in which I would use it, will use it here, is much more general. So here, I'm going to use the term epistasis to describe essentially any kind of interactions between the effects of different mutations. And I'm going to say more precisely what I mean by that. So it's a sort of generic term for genetic interactions. At least I should say in some communities it is used like this. So I had an interesting experience recently where we tried to publish a paper on the biology of aging, and we were thinking about the interactions between different mutations that affect longevity. And the editor of this journal was adamant, saying you cannot call this epistasis. Epistasis means that one thing is masked by another. You should call it interaction. So we had to change the whole paper, replace epistasis by interaction in about 50 different places, because otherwise they wouldn't have published it. So in that sense, this is this sort of difference in different communities. And there's an interesting. So if you want to read more about the history of this of this term, I can recommend a paper, Nature Reviews Genetics by Phillips, in 2008. And he has a very nice quotation about the confusion that is. It's also a little bit depressing. So what he says here is, using the same word to describe subtly different phenomena has generated surprisingly little confusion in the literature, mostly because of the tendency for different areas of genetics to almost completely ignore one another. So this is, but I will use it in this general term, and there's also there's a second quotation that I wanted to mention, from a recent paper by Dan Weinreich, who expresses this general term rather nicely. He says, colloquially, epistasis can be regarded as our surprise at the phenotype when mutations are combined, given the constituent mutation's individual effect. Right? So it's sort of, you have some knowledge about constituent mutations, you make some prediction on the basis of that, and if this prediction is not valid, then there is epistasis. Okay, so this is, of course, so maybe let me write this down here as well. So Weinreich, 2013. This is sort of the surprise at the phenotypic effect of combining mutations. Now this, of course, means that whether you have epistasis or not might depend on your expectation, right? So this is why again, we need a kind of more quantitative description of this. So this depends on your expectations, if you will.
I have one question. So,
Hi, by the way.
Hi. Yeah. So here, in this formula, it would seem that is it the case that once it exceeds 1 pi, the value of pi, then it's 1 for all.
Nope.
It would seem that whenever s is greater than 0, it's always exceeds.
No,
it's actually negative or something. Is there a minus sign missing?
No, it
No?
never exceeds 1, and it never becomes negative.
Is there there must be a because otherwise it's pi is negative.
I'm sorry. Yes.
Oh, okay.
Okay. Thank you. Okay, now it's correct. Good. Right. Okay, so it depends on your expectation. So let's first look at the case of pairwise epistasis. So we have two mutations. So let's suppose we have 3 types, okay? So we have a wild type, F0, and we have 2 mutant types. So this is the fitness F0, mutants with fitness F1 and F2, okay? And so we have selection coefficients S1, which is F1 minus F0, and S2, which is F2 minus F0. Okay. Now the question is, what do we expect for the fitness of the double mutant? Now, what the non-epistatic case, what it's supposed to express is that the effect of the mutation is the same irrespective of where it occurs, right? So mutation effect, so the non-epistatic case means so the mutation effect is independent of background. So in particular, it should be independent of the initial fitness, right? And so here, because we're using this additive setting, the effect of the mutation is that it adds S1 to the fitness. And so if this occurs independent of what the initial fitness is, then this means that the non-epistatic expectation, which I will call F1, 2 with a 0, so this is sort of the expected fitness of the double mutant, is just S1 added to the fitness of F2 of the second type, or, which should be the same thing, S2 added to the fitness of F1 of the first type, and so this would be S1 plus S2 plus F0. Right. So this would be the non-epistatic interaction under the additive model. And so we can express epistasis as a deviation from that expectation. I will call this epsilon 1, 2. So this is the epistasis between the two mutations. And so this will now be, and maybe, you know, it probably makes more sense to express this in terms of the actual fitnesses. So it's F1 plus F2 minus F0, because we have two F0s here. And so the epistasis I now define as the actual fitness of the double mutant minus the expected one, right? And this is then F1 plus F2. F1, 2 plus F0 minus F1 minus F2. Okay, so this is our definition of pairwise epistasis. And here I want to make a remark with regard to what I was saying before about additive and multiplicative scales. If you now do the same argument under the discrete time model or the multiplicative scale, then your picture of what the mutation actually does is that it multiplies the fitness by something, right? So on the multiplicative level, what you would expect is that the fitness of the double mutant, which I now call W12, is equal to the fitness of the wild type multiplied by the two factors that are associated with the individual mutation. So the first mutation multiplies fitness by W1 divided by W0. The second mutation multiplies it by W2 divided by W0. And this is multiplied by W0. So here the non-epistatic interaction the non-epistatic prediction would be this. And of course, you can again see if you take the logarithm, you would end up with this additive formula. Okay. And why is this important? So here, of course, what I was telling you in the additive case, the selection coefficients simply add up. Now here, if you in the multiplicative case, if you were to define selection coefficients as is often done, if we define selection coefficients here now, so you take the ratio of the two fitness values and divide and subtract 1. So this is sort of a common way of defining selection coefficients in the multiplicative setting. Then if you sort of plug this into this equation, you will find that in this case the selection coefficients are not additive. Then these are not additive. Under this equation here, so let me give it a name, let me call it star. So instead what you find is that the selection coefficient, the sort of expected selection coefficient for the double mutant will be equal to S1 plus S2 plus S1 times S2. So this is a little calculation that you can do. And of course, it's, this term will be very small if both the S's will be small. So if the mutations are a weak effect, then this difference doesn't make much of a difference. But generally, I just wanted to mention this here to emphasize that when you look, you have to be careful how you define your scale, and you have to sort of think about what your non-epistatic expectation really is. And this is a theme that I will return to as I go along. Okay? Now the direction that I want to take now is to generalize this a little bit. So one thing that has happened in recent years is that people have become more and more interested in epistatic interactions beyond pairwise, right? So what can one say about higher order epistatic interactions? And to deal with that, you need a mathematical formalism. And this is basically the content of one of the papers that I put into the online material, the paper by Polvik et al., which discusses this, and this is what I want to explain to you a little bit now. And so it turns out that there are sort of two related approaches to describing, yeah.
This epistatic interactions, is this applicable just to, for example, different alleles of a gene, or is it different interaction of two different genes also? I mean,
this could be, this would, I think, applies very broadly. I mean, you can have, any two mutations that you can combine, right? They could be in the same gene, they could be in different genes. This doesn't really matter. So the main point is just you have something that you can combine, right? You have two mutations that you can put into one organism. Okay, so there are sort of two related mathematical frameworks that have been used or propose to generalize this to an arbitrary number of genetic loci. And I call them, and this is actually a wording that goes back to an old paper by Weinberger from the 1990s. I like to call these Taylor and Fourier expansions. Now, this might be a little bit, foreign to you, but I hope you will understand what I mean. And so the first one, so what I call the Taylor expansion. So first of all, does everybody know what a Taylor expansion in the usual sense is? You know how to Taylor expand a function? Okay, I think you can actually ignore that for now, because I'm going to explain to you what I mean by it here. And maybe we can do some, examples in some later tutorial session or something like that. But I think, okay, so you shouldn't, except I don't really have a better name for it. So, just take it as a definition for now, okay? And people who know what it is might. Okay, so let me call it Taylor expansion. So this is, in fact, in some sense, the straightforward generalization of what I did here. Okay. Of the pairwise case. So the first thing that we do is that we formalize this a little bit. So we have in the pairwise case we have altogether 4 types, right? The wild type, 1, 2, and the double mutant. And now I'm going to encode these by a binary variable. Which I call tau, and this tau has 2 components, tau1 and tau2, and these taus can be zeros or ones. Okay, so each of these taus is either a 0 or a 1. And so what I do is I put, so both zeros means that I have no mutation, this is a wild type. 1, 0 is my mutant 1. 0, 1 is my mutant 2, and 1, 1 is my mutant 1, 2, the double mutant. Okay? So this way I can just represent the same thing by these numbers. And then I can write down the fitness of a general, of any type. A general type. Call this now f of tau, so there's now a function of these 2 variables, and this is given by f0 plus s1 times tau1 plus s2 times tau2 plus epsilon 1, 2 times tau1 times tau2. Okay? And you can easily check that this works, so if all the taus are 0, I get f0, which is what I want here, right? If I then put a 1 here, I get an S1. If I put a 1 at the second side, I get the S2. And if I have both of them together, then I also get this epsilon 1, 2, right? But only if both of them are there together, right? So this is sort of the same thing. So this one will get F1. Okay, I don't have to write that down. Okay, so this is the idea. And so this is actually already now the simplest example of a fitness landscape, right? So this is a fitness landscape. Defined on 2 genetic loci. And those of you who know what a Taylor expansion is, you see that this is sort of an expansion in powers of these taus, right? So I have one term of order with no taus, I have linear terms, I have quadratic terms, okay? And so this is now easily and rather obviously generalized to many loci, and this is what we want to do. So now we consider not having just 2 mutations, but having L different mutations, L loci or mutations. And so then my tau is a sequence that now has L elements. Again, these are binary variables. And I can write down a general fitness landscape in the following way. So this is now a function of these L zeros and ones. I have a constant term. I have the linear terms. I will still single out the linear pieces because those are the ones that are non-epistatic. So here, each of these mutations will have a selection coefficient, and it will be multiplied by its corresponding variable. And then I have a lot of other terms, and these I will write in the following way. So here I expand up to order L. So the biggest product that I can have is the one containing all the taus, right? So from pairwise all the way to L. And here I have to sum over all possible subsets. Now in the binary case, in the case of 2 loci, there's only 1 pair, right? Now suppose I have 3 loci, then of course out of 3 loci I can form different pairs, right? I can form 3 different pairs, and if I have 4 loci I can form even more pairs. So what I have to sum over here are all subsets of the set of loci of size k. So this is sort of I1 up to Ik, a subset of loci, which is a subset of all the loci, which goes from 1 to L. So this is a sum over all the subsets of size k, and each of these subsets gets a coefficient, which I call epsilon k, with an index describing the subset. And then I have here the corresponding product of variables. So this is so we have pairwise, triple, and so on, all the way to L. Of course, once I get to k equal to L, there's only one subset, right? So there will always be a term of highest order which contains all the taus, which gives the effect that is only present if all the mutations are there at the same time. Now so now we can think about how many coefficients do we actually have here. So basically what we're doing here is a kind of linear transformation of the fitness values onto these coefficients, the SIs and the epsilon Ks, and this is a linear transformation. The question is it, do we lose any information? And in fact we don't. So how many coefficients do we have? So we have one coefficient of order 0. We have L coefficients of order 1. And here we have to know how many subsets there are of size k, right? So how many subsets of size k can I form out of these numbers from 1 to L? This is now a question to the audience. Sorry? L choose k, right? So we have L choose k coefficients of order k. And so the total number of coefficients is a sum from k equal to 0 up to L choose k, which is 2 to the L. But this is also the number of fitness values, right? Because initially, when I define my fitness landscape, I have to give one fitness value for every combination of these loci, and there are 2 to the L of them. So this tells us that this mapping is actually 1-to-1. So we can always map from the fitness values to the coefficients, and we can go back, right? So the mapping from fitness values epistasis coefficient is 1-to-1. But in fact, for various reasons, this is in fact not the most useful way of defining or of finding higher order epistasis or to quantify higher order epistasis. So therefore, I want to describe a variant or a slightly different formalism, which is what I call the Fourier analysis or Fourier expansion. Now, the argument why we need this is a little bit formal, but the point is that the way this formalism is set up, we cannot, strictly speaking, cleanly statistically separate different orders of epistasis, and this is related to the fact that this expansion here is not an expansion in orthogonal eigenfunctions. Now, this is probably again something that a lot of people will not, will not be useful for me to say for a lot of you. So, who knows what orthogonal? Who doesn't know what orthogonal eigenfunctions are? Yeah, right. Okay. So, let's see. Excuse me. Right, I haven't described the operator. So in some sense, it actually doesn't. So let me sort of try to explain it without using those terms for now, and then I think then we can sort of in the end we can see why it's useful. Okay. Let me just write this sort of as a problem here. This expansion. As defined does not allow for a clean separation of different orders of epistasis. Okay, so what we're going to do is something that initially looks very innocuous. We're just going to say we do the same thing as before, but instead of choosing these taus to be 0 and 1, we choose them to be plus and minus 1. Okay? So to improve this, we're going to encode genotypes by sequences, so still binary sequences. I call these variables now sigma. So sigma 1 to sigma L. And sigma I is simply 2 tau I minus 1, which is now either 0, either minus 1 or 1, right? So you see that when tau is equal to 1, then sigma is also equal to 1. When tau is equal to 0, sigma is equal to minus 1. Okay, so we're sort of replacing the sequences of zeros and ones by sequences of plus and minus ones. Yes. Okay, so what we would like to know, and you know what I'm going to show to you when we talk about the empirical landscapes, we want to have some sort of a measure, you know, how much third order epistasis there is compared to second order epistasis, for example. All right, so we want to be able to somehow quantitatively separate these different orders. Okay, and this is something that is sort of difficult to do with these coefficients. Okay. Okay, so this is actually, you know, this is sort of a trivial exercise. We can just use these new variables. We can plug them in here and we can see what kind of landscape we get. Okay, so let's look again at the two locus case. Two loci. So now we have a function which is now a function of sigma 1 and sigma 2. And this will now have a coefficients, which I will call A. So there's an A0, A1, sigma 1 plus A2, sigma 2, A1, 2, sigma 1, 2, sigma 2. Okay, so this you can still do. Now, what are the coefficients? Let me write them down explicitly. So A0 is equal to F0 plus 1/2 S1 plus S2 plus 1/4 epsilon 1, 2. A1 is equal to 1/2 S1 plus 1/4 epsilon 1, 2. And A1, 2 is actually just proportional to epsilon 1, 2. This is clear because the only way you can get a product of two sigmas is by having a product of two taus. But you can see if you sort of insert the taus here, insert the sigmas for the taus, you sort of generate linear terms as well, right? So you sort of, this is why you see that these coefficients are sort of mixed up here. Now, what are these coefficients actually? What do they actually tell us? So here, we have a clear interpretation of the coefficients. We know that this is the wild type fitness. These are the single mutational effects and so on. Here, this is no longer true. So what is the interpretation? Now, and this is sort of explained again. So here I want to again refer to this paper by Polvik that I mentioned in the reading material. So it turns out if you sort of look at these coefficients, what you find, what they actually express, they in some sense express the same thing as these coefficients, but in a way that is averaged over all possible genotypes, okay? And this is sort of an important point. And this we can easily check in an example. Okay, I should probably, okay, we'll have to do without it. So what is the interpretation of these coefficients? So if we look at the first one, for example, but I think I can still see it, right? Okay, so let me rewrite this a little bit. So this is equal to F0 plus F0 plus S1 plus F0 plus S2 plus F0 plus S1 plus S2 plus epsilon 1, 2. This should be right? So we have 4 times F0, 2 times S1 and S2, and 1's epsilon 1, 2. And what is this? This is simply the mean fitness of the whole, taken over the whole landscape. This is F1, this is F2, and this is the double Newton fitness, F1, 2. Okay? So A0 is just the fitness averaged over the whole landscape. Okay? Now, what is A1? You can do a similar analysis for A1. And what you then find is that A1 is the effect of the first mutation averaged over all possible backgrounds. So what does this mean? So this is a selection coefficient of the first mutation averaged over all backgrounds up to a factor. So what does this mean? So what do I mean by averaged over all backgrounds? What I mean is that in this two-locus system, the first mutation can either occur on the wild-type background, or it can occur on the background where the second mutation is already present. Okay, so there are 2 backgrounds in this case. And I'm going to use the old notation for those. So there's the 0, 0 and the 0, 1 background. And so turns out that A1 is equal to 1/4, so there's some scaling factor here of F. So the first effect that we have is F1 divided by F0, right? This would be the selection coefficient on the wild-type background, and the selection coefficient on the background where F2 is already present would be, okay, let me try to be a bit more precise here. So S1 is the selection coefficient on the wild-type background, and on the background where the second mutation is already present, the effect would be S1 plus epsilon 1, 2, right? And this is exactly what you have here, I hope. Right, so this is sort of the effect on the 0, 0 background, and this is the effect on the 0, 1 background. And this is sort of true generally. Okay, so all of these coefficients now describe the effect of the mutations not on the wild-type background, but averaged over all backgrounds, which is a bit less intuitive. And this is, again, for those who know what Taylor and Fourier expansions are. So a Taylor expansion of a function typically means that you pick a reference point and then you try to describe your function close to that reference point, whereas a Fourier transformation is something that describes a function globally. And this is very similar to the difference between these two approaches here. Okay, and so again, we can do this for any number of loci. Loci. And again, it's just, it basically, the formula looks the same as before, except that I now use the sigmas instead of the taus. And I'm going to write it, so here, it's not that important to somehow single out the zero and first order effects. So I'm just going to write it as one big sum from order 0 up to order L. And then here again, we have the sum over the subsets, where now the subset can also be empty. And here then we have some coefficients, a I1 up to ik, and a product of sigmas. So this is sort of the general formula for an L-locus fitness landscape in this formalism. And what you should notice is that these things here, these products here, so these are products of pluses and minus 1. So the only value that these products can take are plus and minus 1. So in this formalism, each fitness value is obtained from all the coefficients, right? So all the coefficients enter here, just with different signs. So the way in which different sequences or genotypes are distinguished are just by the signs in front of the coefficients, but all the terms always appear, which is very different from the expansion that I described to you before, okay? So every fitness, every fitness value is a sum. So these are now what I call Fourier coefficients, right? So these are Fourier coefficients. I should say that there's a simple formalism in computer science, and there these are called Walsh coefficients. So in the literature, you will find both of these terms. So every fitness value is a sum over all coefficients with random signs, or no, not random signs, with alternating signs. I mean, the signs can be plus or minus, and the sequence of pluses and minuses changes. And so in that sense, this is a very global description. So every coefficient depends on all the Fourier, on all the values. Kind of global description. And the key property of this expansion is, as I mentioned in the beginning, that it's an expansion in orthogonal functions. And this is, if you want to, want to learn more about this, there's a problem. So the problem set that I posted on the web page is about that. This is an expansion in orthogonal eigenfunctions. And you ask what the operator is. So the operator is a so-called graph Laplacian of the sequence space, okay? Orthogonal eigenfunctions of the graph Laplacian. And this is the topic of problem 2, this problem sheet. I think it's maybe best, since this is not I think not that important for what I want to go on doing, that, you know, if people are interested in this, maybe we can sort of organize a separate session where we talk about this aspect. I mean, this is sort of where this expansion in some sense comes from, but for using it, it's actually not that important. Okay, so maybe we can come back to that a little bit later. So before, I think my time is essentially up. But let me just write down, you know, the important result that follows from the fact that we have this orthogonal expansion, because this is sort of what we'll be using later. And it comes back to this problem that I mentioned in the beginning, that you would like to be able to decompose your fitness landscape into contributions of different orders. So the consequence of this is that if you write, and this is actually, I think, I mean, I never really got to the heart of this, but I think this is very similar to what is called the ANOVA analysis in statistics, which I think many of you are familiar with. So the idea is that you can decompose the variance of the fitness. So let me just define it like this. So the variance of fitness, what do I mean by this? I just mean the sum over all the genotypes. So you take the fitness of your genotype and you subtract the mean and you square it, right? So this is sort of the overall variability of fitness. And because of this property that these things are all orthogonal to each other, you can write this in the following form. So you can write it as a sum over orders from 1 to L, where 1 is the linear order. And then each term is just the sum over the squares of these coefficients. And here again, there is a sum over the subsets of size k. Right. And this is now, if you will, the contribution of order L. So this is a contribution of order k. Sometimes also called the Fourier spectrum. And so for k, and so for k equal to 1, this is the linear piece, so the non-epistatic part. And for k greater or equal to 2, these are the epistatic contributions of different orders. And nowadays, when people talk about higher order epistasis, this typically means higher than pairwise, so k greater than 2 would be what is called higher order epistasis. Okay, I think this is a good time to stop. So thank you for now. So tomorrow I'll be talking about the sort of the second important aspect of epistasis, which is called sign epistasis. This refers to this, to the paper by Dan Weinreich and collaborators that I also posted on the webpage. Okay, thank you very much.